Hello, my name's Adam Smith and I'm a postgraduate researcher at the University of Exeter Medical School. This video describes the practical application of pyrosequencing to analyse a specific amplicon or DNA fragment in terms of its DNA methylation. As with all forms of sequencing, the DNA fragment of interest must first be amplified by polymerase chain reaction or PCR. In this case, bisulfite treated DNA is used to enable the measurement of cytosine methylation. For information on bisulfite treatment, see the video below. The bisulfite treated DNA is added to a cocktail of water, nucleotides, buffers, a polymerase enzyme, and primers for the target region. These primers are designed to be specific for the bisulfite treated DNA of the target region and contain a biotin tag that we'll come back to later. A no template control, or NTC, and a fully methylated control is usually also included. Again, we'll come back to what these are used for later. The DNA reagent mix is then placed on a thermocycler to amplify the targeted DNA. This can take anywhere up to 3 to 4 hours. After the amplification of our target region is complete, we need to check that everything has worked up to this point as we expected. To do this, we load the DNA onto a 2% agarose gel and perform gel electrophoresis. This is a typical gel image of what we would expect to see after PCR amplification. As you can see, for each sample labelled 259 to 268 and the methylation positive sample, we have a bright white band visible. The ladder on either side of the gel enables us to estimate the size of the gene fragment we have amplified. This amplified region is meant to be 246 base pairs in length, and as you can see, the band we can see is sitting between 200 and 300 band on the ladder. So we can be quite confident that this is the product we want. The no template control, or NTC, has no band at all. This is good as it means there is no contamination in our PCR. These samples can now be taken forward to pyrosequencing. 20 microliters of the well-optimized PCR product per sample is needed for the pyrosequencing reaction. Each sample is mixed with 2 microliters of well-mixed streptavidin-coated cephorose beads, 40 microliters of binding buffer, and 18 microliters of high-purity water. Each 80 microliter sample is then added to a well of a 24 well plate and placed on a shaking station for 10 minutes. The biotin tag we added during PCR binds to the beads at this stage and enables us to clean the PCR product in the next step. At the same time, a sequencing primer specific to the PCR product gets diluted in a annealing buffer to a final concentration of 0.3 micromolar. 25 microliters of the diluted primer is then added to each well of a Q24 plate and then put to one side for later. While the DNA is mixing, we fill the wash station troughs with water, ethanol, denaturing solution and wash buffer. Using the vacuum tool, we suck up the total volume of water in the second water trough to clean the filter probes, replacing the water once we've finished. Once the 10-minute mixing has finished, we can place the DNA plate and the Q24 plate containing our sequencing primer on the wash station, making sure it's the right way around. And we're then ready to begin. Again, using the vacuum tool, we suck up the well-mixed 80 microliters of amplified DNA plate, flipping it upside down for 15 seconds to ensure the entire sample is bound to the beads is collected on the probes. We then move the vacuum tool through the ethanol for 5 seconds, the annealing buffer for 5 seconds, and then the wash buffer for a further 10 seconds. Raising the vacuum tool to 90 degrees for 10 seconds allows the liquid to drain from the filter probes. Turning the suction off and slowly bringing the filter probes down into the waiting Q24 plate containing the sequencing primer. We then shake the tool gently from side to side, releasing the beads into the solution. The Q24 plate now needs to be heated on a heat block for 2 minutes, but no longer, otherwise you risk evaporating your sample. Following this, the samples can be loaded into the pyrosequencer and left to cool to room temperature for 5 minutes. During the 5-minute incubation, we can prepare the sequencing cartridge. This is filled with modified nucleotides, A, G, T and C. We also add the enzyme and substrate mixes to the cartridge. Once this is complete, it can also be loaded into the pyrosequencing machine. Once the Q24 plate has cooled to room temperature, we can start the pyro run. Click the link below for more details of the chemistry of the pyrosequencing reaction going on within the machine. The pyrosequencing has now finished, and we can have a look at the results. As you can see, for each sample we have a light emission trace corresponding to the incorporation of a specific base into the target sequence. Using the software, we can analyse the data to get a measure of DNA methylation at each of the 4 sites in this region. This sample has worked really well, with all sites having high sequencing quality. However, this sample seems to have a loss of quality in the final 3 sites, and so it will need to be excluded or repeated. We can again see that the NTC has not produced a trace in the pyrosequencer, confirming that no contamination has taken place. And finally, the methylation positive sample we use as a control has shown methylation values of above 90% at every site. So we can be confident that the process has worked successfully, and we can use these results in our analysis of pathology.